Everybody, Dr. O. Let's review the lymphatic and immune systems, okay? Now, some textbooks like to separate the two, but I really think they fit really well together. All right, what makes lymphatic fluid different than blood plasma? So blood plasma is water, but 92% water, but it also has a lot of plasma proteins. So when blood plasma, the fluid portion of your blood, is filtered out into the space between your cells and becomes interstitial fluid, the plasma proteins get left behind. They're too big. So most of that fluid is going to be sucked back in. The numbers that I use are that 24 liters of fluid is filtered out every day. 20.4 liters is sucked back in or reabsorbed by osmosis because of those proteins. The other 3.6 liters is interstitial fluid, but then becomes lymphatic fluid once it enters a lymphatic vessel. So the main difference is the plasma proteins. So really, interstitial fluid and lymphatic fluid are the same thing, just in different places. Is the fluid still between the cells, or has it made its way into a lymphatic vessel? All right, lymphatic vessels are not circular like the circulatory system, so we don't have arteries going away and veins coming back. They begin with terminal lymphatics. They all begin out at the ends and they work back towards your heart. Okay, are lymphatic vessels more like arteries or veins? It's veins. They're thin-walled, large lumens. They have valves, or something like valves anyways, so much more like veins than arteries. Are the quote-unquote valves in lymphatic vessels really valves? Do they still behave like valves? Well, kind of answers the question. We'll call them valves, that's fine. They're not technically valves like you see in the heart or in veins, but they're overlapping endothelial cells. I like to think of them more like a trapdoor than anything. Once fluid or a bacteria or anything else falls through one of these flaps, they can't get back out. So they function like valves because they prevent the backflow of material. Good thing about that is, let's say you are a bacteria, you fall into one of our lymphatic vessels, you can't turn around and get back out. You have to go forward, which means you're going to have to go through lymph nodes and lymphatic organs and all the other structures, and hopefully our immune system will find you, remember I'm talking to the bacteria now, and stop you. Okay, which parts of the body drain into the thoracic duct or the right lymphatic duct, and where do they both drain into? So the right lymphatic duct is going to be the right side of the head, right side of your thorax. The thoracic duct is going to be everything else. So the right lymphatic duct, right half of the head, right thorax, right arm. The thoracic duct, everything else, left side, and then the lower extremities. Where they both drain into, they both drain back into your subclavian veins and into your circulatory system, back to your heart. So that 3.6 liters of fluid that was filtered out at your capillaries but not reabsorbed is going to work its way back through your heart, to your heart. But before it gets there, it's going to be filtered and cleaned, and your immune system is going to recognize things that shouldn't be there during that process. That's the whole point of having a lymphatic system. All right, why are they called T cells and B cells? Well, it has to do with where they mature. So T cells are thymus dependent. They depend on your thymus gland. B cells are bone marrow dependent. So obviously both very important. We'll cover different parts here. All right, with T cells, what role do the cytotoxic T cells play? Some call them killer T cells. Well, I like cytotoxic T cells, or CTLs, helper T cells, and suppressor T cells. Suppressor T cells are also, they're more often now you're hearing them called regulatory T cells or Tregs, but these cytotoxic T cells, they're your killers. Once they've been activated, they're involved in cell-mediated immunity. They will directly attack and try to destroy cells that shouldn't be there in foreign invaders. Helper T cells, we call those the generals, because they activate your cytotoxic T cells, but they also activate B cells. So I call them the generals because they're not getting their hands dirty, but neither arm of your immune system, your T cells or B cells, would work without it. Your suppressor T cells or your regulatory T cells, their job is to make sure the response isn't too big. Think about an allergy. If someone has an allergy, that's a malfunctioning immune system. No one should eat a peanut and drop dead. That's what happens if the immune response is too aggressive, it's too big. So your suppressor T cells or these regulatory T cells, they should stop that from happening. All right? So cytotoxic T cells, think directly attacking things that shouldn't be there. It's called cell-mediated immunity. Helper T cells, they're the generals that regulate and control other T cells and B cells. Suppressor T cells or regulatory T cells, they keep the response from being too aggressive, or at least they should. That's why people that have more regulatory T cells or suppressor T cells will be less likely to have autoimmune diseases, asthma, etc. All right, B cells become plasma cells. What do plasma cells make? They make antibodies. So when a B cell is activated, it becomes a plasma cell. Plasma cells make antibodies. So we'll cover antibodies more later. But I think of, and this will tie into other questions, but your T cells, I think of them as knights with a sword or a battle axe. They're directly attacking, destroying cells that we don't want around. B cells are the archers. So they make the antibodies that flood the fluids of our body with these antibodies, that they either cause cells that shouldn't be there to clump up, or they target these cells for destruction so your other immune cells can see them, this type of thing. So I think of them as more the archers. They're staying back but producing something that's actually doing the work. So B cells become plasma cells make antibodies. All right. Excuse me. Natural killer cells are primarily involved in blank and allows them to attack and hopefully destroy cancer cells and virus-infected cells. That's called immunological surveillance. So natural killer cells, the key term with them is immunological surveillance. They're looking for cells that have abnormal features on their surface. So once a cell's been infected by a virus, it should put like flags on it saying there's something wrong, or the fact that it's producing different products. Same thing with cancer cells. They should change. So natural killer cells can't see all of them, but natural killer cells are very, very important in the immune response, especially in those two areas, destroying precancerous and cancerous cells as well as virus-infected cells. So, all right, what is MALT? MALT stands for mucosa associated lymphoid tissue. So think anywhere where you have a lot of mucous membranes, the lymphoid tissue here is going to be called MALT. Digestive system, respiratory system, reproductive systems, urinary system, all these areas. So specific other places you're going to find MALT, I'm trying to think of any you might have to know by name. You have Peyer's patches. So in some areas of the intestines, you have these really concentrated areas of MALT, mucosa-associated lymphoid tissue, called Peyer's patches, P-E-Y-E-R-S. But the main two you're going to have to know is your tonsils and your appendix would be MALT, mucosa-associated lymphoid tissues. All right, next question here. How many tonsils are there? We have 5. So you actually have your pharyngeal, which means throat, your 1 pharyngeal tonsils. That's the one we call your adenoids. You have 2 palatine tonsils in the area of the palate, and 2 lingual tonsils in the area of the tongue. So yeah, you actually have 5 tonsils. So the 1 adenoid, your pharyngeal, a pair of palatine, and a pair of lingual. All right, lymph nodes are filters. What percent of antigens are filtered out? 99% or more, right? So they do a phenomenal job of this. As lymph is traveling through your lymph nodes, it's being filtered and cleaned. And also, if things are found that shouldn't be there, it's being taken to your immune system. So it's going to be recognition or antigen presentation, which is the next question. What is the first step of your body's immune response? Remember that you cannot fight an enemy that you do not know is there. That's this term antigen recognition. When a macrophage, eats something, or when something gets caught in a lymph node, the first thing we do is we present it. We present it to the other immune cells, and we say there's these antigens here that shouldn't be here. So we recognize there's now something there. That's when you can create immune response against it. So antigen recognition and then antigen presentation, presenting it. There are special cells that present antigens they find to the rest of your immune system. Most importantly, your helper T cells, so they can actually activate an immune response. Okay, the thymus gland plays a key role in the maturation of the immune system. What happens to the thymus gland as you reach puberty and beyond? So, the thymus gland is huge when you're young. So like as you're a developing fetus, and when you're young, it's large, and weight-wise, it's probably largest when you hit puberty. But versus the rest of your body size, it's larger than that when you're young. But its job is to help your T cells mature. So the reason we have a robust, active thymus gland when we're young is because we're building a new immune system. So what actually happens to it as we get older, that's a problem, right? Our thymus gland shrivels up and becomes fibrous, so it's less effective when we're older, which is why people that die from infectious diseases are either really young, because they have an immature immune system, or really old. We don't have a good immune system. So at puberty, let's say your thymus gland weighs 1 to 1.5, like 1.3, 1.4, 1.5 ounces. By the time you're 50 years old, it's probably going to be 0.3 ounces or less. So it's basically shriveling up and becoming fibrous. And that's why when you get older, you don't have this robust immune response. All right, so that's what happens to the thymus gland. It becomes smaller and more fibrous, and its role is the maturation of your T cells. That's why they're called T cells, thymus dependent. Okay, what is the function of the red pulp of the spleen, or red pulp portion of the spleen, and the white pulp portion? So, won't go into huge detail here, but the spleen is really two organs in one. It's an organ that plays a role in keeping your blood clean, and then it's an immune organ. The red pulp portion of the spleen is all about filtering damaged and old red blood cells. So it plays a big role in your red blood cell maintenance. That's why it's red, because when it takes on a red blood cell and breaks it down, it stores the iron, which is what makes it red. So red pulp, or red portion of the spleen, think red blood cells, circulatory system. White pulp, it's like a huge lymph node. So it's got all your immune cells are there, so it's monitoring what's going on from an immune standpoint. So the white pulp portion, think white blood cells, think immune system. All right, next, what is the difference between a specific and a non-specific defense? So a non-specific defense doesn't care who you are. Might not even know you're there, right? So skin is a non-specific defense. It's part of your immune system. It's keeping things out, but it's not choosing what it keeps out. It's just a barrier. Stomach acid is the same thing. These are examples of non-specific defenses. Fevers. Specific defenses, they're going after a very, very specific target. So that's going to be your specific immune system versus what we call your innate immune system. All right, what kind of white blood cells are involved in the specific defense system? So what we just got done saying, the ones that actually know exactly who their target are, that's going to be your T cells and your B cells. So once they've been activated, I think of wanted posters. Your helper T cell gives them a wanted poster. Here's your target. If you're a T cell, go attack and destroy it. If you're a B cell, become a plasma cell and make antibodies against it. Now, both those T cells and B cells, once they're activated, will also make memory cells, which is why the second time you're exposed to something, or if you've been vaccinated and the vaccine was effective, you won't get sick because the immune response is already waiting for it. So that's going to be the white blood. So the lymphocytes, T cells and B cells, are going to be the key answer there. Next, do interferons play a role in stopping bacterial or viral infections? It is viral infections. They decrease viral replication. So they also stimulate other cells. So they stimulate your natural killer cells and your macrophages, because there are two kinds of macrophages, just means big eaters. They're the kind of cells that will engulf and destroy virus-infected cells. So not only do they slow viral replication to try to slow the spread of a virus, they also tell your immune system to come look for these viral infected cells there. I jotted down, there are a few different types, but I don't ask you to go to this level, but maybe when you get into your other nursing classes or whatever, you might have to know these. The alpha interferons, they stimulate natural killer cells. Beta interferons stimulate fibroblasts, because they can come in and clean up debris and damage. And gamma interferons, they're going to stimulate your T cells and your natural killer cells, as well as macrophages. So I won't ask you to know that, but just be familiar with the fact that there are more than one kind of interferon. Interferons are produced by these virus-infected cells, but they also can be used as a treatment. We can produce them as well. I mean, outside of the body, and use them as a treatment. Okay, what kind of proteins are complement proteins? The complement system complements the action of what? So they are plasma proteins. They're made by the liver and they're in your plasma. So complement proteins are plasma proteins. They complement the actions of your antibodies, number one, and phagocytes. So your complement proteins, a simple way to look at it, they dock on the surface of a cell that they want destroyed, and they can actually burrow holes into that cell, and if you do that enough times, the cell will get weak and die. So they can actually directly kill some cells, but they also, think of them having like a strobe light. So there's a strobe light that basically activates antibodies and phagocytes to come to this area. So if they can't destroy it directly, they'll plant a flag, a strobe light, whatever, a flare, whatever you want to call it, and they help antibodies work better, and also they call phagocytes to the area. These macrophages and phagocytic cells, they can't see, right? So they need something to draw them towards their target, and that's what these complement proteins will do. Okay. What are the 4 cardinal signs of inflammation? English is fine, Latin not necessary. So if you want to know the Latin, it's rubor, tumor, calor, and dolor. That's how I had to learn them. But those terms stand for redness, swelling, heat, and pain. So rubor, redness, tumor, tumor, swelling, calor, heat, and dolor means pain. So redness, swelling, heat, and pain are the 4 cardinal signs of inflammation. If you want to know the fifth, it's a loss of function or inhibited function, usually because it hurts, right? So redness, swelling, heat, and pain, those are your 4 cardinal signs of inflammation. What do mast cells release, and what does it do? So mast cells, along with another kind of cell we covered earlier in an earlier chapter, basophils, they release histamine and heparin. So they used to think they were kind of the same cell. They have slight differences, but mast cells and basophils release histamine and heparin. So histamine is pro-inflammatory. That's why if you have allergies, allergic rhinitis or something, you're going to take an antihistamine to stop that. So histamine is an inflammatory compound. Inflammation is necessary for your immune system to fight off infections. The problem is we don't want too much inflammation, and we don't want it when we're not fighting off an infection. When your body senses inflammation, it assumes it's under attack by virus, bacteria, parasite, etc. The other thing that mast cells release is heparin. Heparin's a blood thinner. It allows more fluid to go to an area so more immune cells can show up. So mast cells and basophils both do the same things. All right, what kind of cells produce scar tissue? That would be your fibroblasts. They lay down collagen. So collagen is what forms scar tissue. Notice that there is a fair amount of overlap between this chapter and the chapter on blood cells. For example, T cells are involved in cellular-mediated or cell-mediated immunity, and B cells are involved in humoral or antibody-mediated immunity. Cell-mediated immunity means a direct cell-to-cell contact, direct cell-to-cell attack. Humoral means body fluids. So your antibodies are dumped in body fluids, and that's when how they go do their job. So that's something we covered back in this chapter on cells. We also covered the basics of the neutrophils and eosinophils and all those as well. All right. Know the different types of immunity, so passive versus active, innate versus acquired, etc. So let's go through the 4 main types. Just real quickly though, passive immunity means you're not doing anything, it's being done to you. So I'll give you some examples there. Innate means you already have it, you're born with it, it's genetically determined. Acquired means you have to have been exposed. So so we have natural active immunity. That is, you're exposed to an antigen, which whether from an actual infection or from a vaccine, you're exposed to an antigen, your body actually builds an immune response. That's the robust, natural active immunity response. Artificial active immunity, so there is going to be an immune response, but it's not actually, you're not doing it. That's going to come from breast milk. So breast milk has all 5 classes of antibodies in it. So mom did the active work. Mom produced the antibodies. She's just giving them to you. So that's artificial acquired active immunity. Natural passive immunity is going. I think I got myself screwed up there. So naturally acquired active immunity is you get exposed to an antigen, you respond to it. Artificially acquired active immunity would be the vaccine, where you are exposed to this artificial antigen that's not going to make you sick, but you'll still develop immunity to it. Naturally acquired passive immunity, that would be mom's breast milk. The antibodies coming from mom, so it's a natural way of doing it, but it's still passive immunity. You, as the baby, are not making the antibodies. Sorry for that, I confused myself. And then lastly, we have artificially acquired passive immunity. That would be antibody therapies. Like if you got bit by a snake, they'd give you a snake antivenom, or like they can treat Ebola with antibodies that they give you. So you're not making the antibodies, you're acquiring them in an artificial way. These are going to be for infections where you wouldn't have time. You wouldn't have time to, by the time you could develop antibodies against the Ebola virus will have killed you if it was going to kill you. So apologize for the confusion there. So passive immunity, it's being done to you. Active immunity is going to be the robust immune response coming from you, not your mom or not a drug. Innate, you're born with it. Acquired, you acquire it. All right. What is the purpose of memory cells, both memory T cells and memory B cells? It's memory. So now this isn't perfect, but in a perfect world, if you're relatively young and healthy, you have memory T cells and memory B cells that are looking for every foreign invader that they've ever come into contact with. This is where the secondary immune response comes into play, and this is why vaccines work. If you get a vaccine and it works, or if you got exposed to something and didn't die, you should now have memory cells looking for it. So, and we'll have a specific question about secondary immune responses later. So the next time you're exposed to something, you have immune cells that are waiting. So the response should be much larger and much quicker. So that's the purpose of memory cells. All right, what kind of T cells have CD4 receptors and what kind of T cells have CD8 receptors? So this is important in a couple areas, like with HIV infections. HIV only infects the CD4 positive cells. So CD4 receptors are going to be on your helper T cells, those generals we talked about. CD8 receptors will be on the cytotoxic T lymphocytes. In what ways are helper T cells needed? Do they do more than just help T cells? So the answer is yes. So helper T cells, they activate your T cells, but also activate your B cells. So without helper T cells, you can have T cells, but they're going to be naive. They don't know what to do, or they don't have a wanted poster in front of them. Same thing with B cells. That's why if someone has AIDS, because their helper T cells are gone, they have T cells, they have B cells, but they can't fight off infections because they don't have that general telling them what to do. So they activate or sensitize your T cells and your B cells. What do suppressor T cells prevent? I still love that term, but again, every time you see suppressor T cells, remember that a lot of people are going to use the term regulatory T cells or Tregs. So suppressor or regulatory T cells, what do they prevent? They prevent an overactive immune response. You do not want to fight every single antigen you're exposed to death. You do not want massive immune responses all the time. That's what allergies and anaphylactic shock and these types of things are caused by. So the suppressor T cells, they're trying to stop the immune system from being overactive. Then what is their other name? That would be your Tregs, or just so you know, it's capital T, and then you'd see like a R-E-G-S after it, or regulatory T cells. Okay, next. What part of an antibody's structure determine which antibody it is? IgG versus IgA, which we'll get to in just a moment. So it's called the constant segment of the heavy chain. It doesn't really matter, but constant segment is the key. That means that whenever you see that constant segment, you know which type of antibody it is. IgG, A, M, E, or D. The variable segment is what makes it specific. So yes, I'm an IgG antibody because of my constant segment. The variable segment says I'm looking for that specific target. This is my wanted poster. You have a different wanted poster. So just remember that, constant versus variable. Not a huge deal, but worth noting. Okay. The 5 classes of antibodies, how I remember them is GAMED, G-A-M-E-D. So I have them in a different order here. But so the 5 classes of antibodies are IgG, IgE, IgD, IgM, and IgA. So the GAMED, I-G-G-A-M-E-D. Let's look at what each of them do. So they're all antibodies, which means that they can neutralize things, they can cause things to clump up, they can cause things to lyse or tear apart. One of their main functions is calling in other cells to target things for destruction, that kind of thing. All right, let's look at some of the key examples here of the 5 antibody classes, though. Which antibody class plays a key role in allergic reactions? That's going to be IgE. Which is in glandular secretions. That's going to be IgA. These are often called secretory IgAs because they're in all glandular secretions. So mucus, saliva, tears, semen, any body fluid would have these IgA antibodies. Which play a role in the sensitization of B cells? That's going to be the IgD antibody. Which is the most numerous by far is the IgG. Which is the first to show up. It's the IgM. IgMs are these huge structures. They're a pentamer. They look like 5 antibodies fused together. They're huge. That's kind of what they're famous for. And they are the first to show up, but they're not very good. I call them like a stopgap. They're trying to hold off an infection until the more important antibodies, specifically the IgGs, can show up. They just take time to develop. And then which are passed to the infant through breast milk? That would be all of them. So IgG antibodies are the only antibody that make it through the placenta to the developing fetus, but all 5 classes of antibodies are in breast milk. That's why, we were designed and evolved to loan moms to loan their immune system to their babies, and that's why the baby's immune system is pretty immature when it's born, because they were designed to get that help. All right, what are the key differences between the primary and secondary immune response? So 2 big ones. So your memory cells are going to respond much faster. We've already mentioned that. So the primary immune response is slower. So you'll see like the IgM antibodies will show up first, but they're weak. So chances are you're going to get sick. Your immune system is trying to fight off an infection, but the full immune response won't be there for 10 to 14 days, the IgGs and everything else. So it takes 10 to 14 days to mount this first immune response, which is why the first time you're exposed to something, you usually get sick. because while your body's mounting a response, the infection's winning. So the primary immune response is slow, takes 10 to 14 days, and at the beginning anyways is dominated by IgM antibodies, which just aren't that effective. The secondary immune response is much quicker, and it's dominated by the more powerful IgG antibodies from the beginning. So that's why the second time you're exposed to something, you don't get sick. Now you might say, like, wait a minute, I get a cold every year. Well, there are thousands of viruses that cause the cold. You shouldn't get the exact same cold twice, though. Or if you get a vaccine and it was effective, you shouldn't get that disease. So this is not a perfect system, but that's once you have memory to an invader, the second immune response should be much more powerful. That's how I define vaccines. I mean, vaccines do induce immunity, but the way I like to say it is that when you get a vaccine and it works, the first time you're exposed to a pathogen in the real world is the second time your immune system has seen it, and that's why you shouldn't get sick. All right, now obviously we could do a whole long thing about this, but what are the key issues with allergies, autoimmune diseases, and the immunodeficiency diseases? So allergies, they're a hypersensitivity. Your immune response is way too large. So like I mentioned earlier, no one should nibble on a peanut and drop dead from anaphylactic shock, which we'll get to in the next point. So it's an overactive immune response. Autoimmune diseases, now you treat them with immune suppressant drugs, but that's not what they are. Autoimmune diseases they've lost tolerance. Autoimmune diseases, your immune system is attacking normal healthy tissues. It can't tell friend from foe. So allergies, too strong an immune system. Autoimmune diseases, wrong targets. They're targeting things that are normal and they're convinced they shouldn't be there. And then immunodeficiency diseases, that's a weak immune system. So it's not a strong enough immune system. So you could argue that just getting old is an immunocompromising disease, right? Causing immunodeficiencies. So they're all wildly different. But just know the keys there. All right, last point. What is anaphylactic shock? So shock, when I think of shock, I usually think of blood loss, right? If you lose 20% or more of your blood loss, you go into shock. So that'd be hypovolemic shock, or a shock caused by a low blood volume. Anaphylactic shock is different. Here's what happens. You get this massive immune response, and it causes your airways to constrict. So bronchoconstriction. So now you can't breathe, having trouble breathing. And vasodilation. Your blood vessels all pop open. Problem is, we only have 5 to 6 liters of blood. Blood needs to be, if you're going to be digesting a big meal, blood's going to go to your gut from somewhere else. We don't have enough blood for the entire body to get all the blood flow at once. So we distribute it based on needs. When you exercise, the blood flowing to your kidneys and your guts goes down, vice versa. So what happens there is, if all your blood vessels pop open at the same time, we don't have enough blood. So you are going into shock, not because you're bleeding, but because your blood vessels have dilated. So anaphylactic shock is bronchoconstriction, your airways get small, vasodilation, your airways dilate. So the treatment here would be epinephrine. Epinephrine is a sympathetic agent, part of your fight or flight system, which would cause bronchodilation and vasoconstriction. So it does the exact opposite, and that's how it saves people's lives. Okay, big meaty section, I get it. This is a huge deal. We could easily have broken this down into a lymphatic system and immune system, but this is to me, the connecting them is the best way to go. So I truly hope this helped. Let me know if there's anything I can do to help you. Have a wonderful day. Be blessed.